You will pull this thing. Like presentation. Share. Yeah.
All right, so it's the top of the hour. I'd like to welcome everyone to today's JMCC ISHR cardiovascular webinar. Thank you all for joining. I'd like to thank the JMCC and the ISHR for their continued support and sponsorship of this series, and remind everyone that both the JMCC and JMCC Plus are actively looking for quality submissions in the topics you can see here on your screen. So please check out the website and social media to learn more about special calls for papers and other programs that JMCC and JMCC Plus are offering for you. And so with that, I will turn things over and pleased to introduce today's moderator for our session, Dr. Tish Murphy, to introduce our speakers and this month's Editor's Choice paper, which is the topic of our webinar. Thank you all again. Just a quick reminder, please enter any questions for the speaker into the Q&A box, and they'll be addressed at the end of the session. Thank you.
Hi, everyone. I'd like to also extend my welcome to everyone for discussion of this paper. I'm Tish Murphy. I'm at the NHLBI NIH Intramural Program, and I'll be the moderator for this session. Today, we're going to discuss a paper, Sex-Dependent Phosphorylation of Argonaute 2 reduces the mitochondrial translocation of miR-181C and induces cardioprotection in females. Today's authors who will be here to discuss this paper are Dr. Sam Doss and Dr. Diego Quiroga. And I'll just introduce them a little bit. I've known Sam for a long time. He got his PhD in Debrecen in Hungary, and then went on to do a postdoc at Hopkins. And then he went on to establish his own lab there at Hopkins as well. And he's currently assistant professor in the Department of Anesthesiology and Critical Care at Johns Hopkins. Sam has been very involved in the ISHR. Early on in his career, he was highly involved in the North American Section ECI group, and he's currently a counselor on the North American Section Council. Also joining us today to discuss this paper is the first author, Diego Quiroga, and he's a postdoc in Dr. Doss's lab at the Department of Anesthesiology and Critical Care. And he received his PhD in Buenos Aires in Argentina. And so without further ado, I'll turn it over to Sam and Diego, who will discuss it, and then I'll lead the discussion at the end. And as Ron said, please put your questions in chat or the other option. All right, thank you. Turn it over to Sam.
Yeah, thank you, Dr. Murphy, for the kind introduction. And also, I would like to thank Ron for organizing all of these and the email for Jenny. I really want to thank JMCC editorial team for giving us this opportunity to share our data here. We are very excited about this paper, not only that we identify a mechanism how microRNA can translocate into the mitochondria, but we see a gender differences in these mitochondrial translocation. And total credit goes to Diego, because Diego is the one who pushed me for this study. So let's start with the honoring these two gentlemen, Dr. Victor Ambrose and Dr. Gary Rufko. These two are the one who found microRNA in our non-coding RNA junk trash can. That's what they used to call before 1995, but they identified this tiny little RNA. They have a profound physiological importance. And the way microRNA works, I'm sure that this does not need any kind of introduction, but what I would like to tell you is why we thought this is something different, what we are doing. MicroRNA binds to the 3' UTR of messenger RNA in the cytoplasm. And what it does is they recruit RNA binding proteins such as AGO2, GW182, TRBP1, and TRBP2. And they form this complex, and this complex is called RNA inducing silencing complex, or RISC. And this RISC is the main, or we call it business end of microRNA, and inhibit the translational inhibition, so the protein cannot synthesize. Sometimes we see microRNA degrade messenger RNA. So this was the initial thoughts, and everybody was publishing on this. About a decade ago, we asked this question: can microRNA express into the mitochondrial fraction? The reason we thought it's kind of relevant, because in mitochondrial genome, we see only 13 gene transcribed. However, if we do RNA sequencing or proteomics, we see hundreds, not thousands of RNA or gene or protein. So how they get into this? So not only regular messenger RNA or protein, but we also found 5S ribosomal RNA in the literature. They translocate into the mitochondria. That was the knowledge we had, that microRNA may be similar to other non-coding RNA. They can translocate into the mitochondria. So with encouragement of my mentor, Dr. Steenbergen, and obviously Dr. Murphy, they did not let me down, and they said, yeah, why don't we try it? So we organized a meeting, and we say, we will just check the total heart homogenate and mitochondrial lysate, and we will perform a microarray, and we will see whether any microRNA is enriched in the mitochondrial fraction, and which we did. There are lots of challenges, because at that time we did not know how to isolate RNA from the mitochondrial fraction, because the RNA is different. But anyway, we spent a lot of time, and we are kind of optimized this method of isolating mitochondrial enriched microRNA fraction. So we identified and validated microRNA 181C, highly expressed in the mitochondrial fraction of the heart. And when we published that, not only we showed that microRNA in the mitochondria, but we showed the functionally active. They go inside the mitochondrial matrix, target mitochondrial enriched or encoded gene MTCOX1. And by doing that, it forms a complex for remodeling. And because of that, we see an excessive ROS production. So this was very important to our study, that we not only see a microRNA is expressed, like other published paper at that time, but we showed it's functionally important. And then what we did is we've checked for all those other paper whether anybody can do that. After several years, like 3 or 4 years, a group from Cleveland Clinic, they used Rho0 cell. In these cells, there is no mitochondria. And then they use the regular cell and Rho0 cell, mitochondria DNA less cell, and they perform next generation sequencing. And they also identify microRNA 181C is missing in Rho0 cell and present in this mitochondria in present cell. So therefore, independent validation of 181C in the mitochondria. We also overexpress this microRNA 181C in vivo using liposomes. And after 3 weeks, we see a statistical significant difference in both ejection fraction and fraction shortening. And you can see the dilation of the cell cardiac echo data, therefore suggesting overexpression of microRNA 181C can induce heart failure. So at that time, we had this mechanism in mind, that when microRNA 181C goes up, it directly binds to the 3' UTR, 3' end of MTCOX1. Therefore, COX1 protein goes down, and we see excessive ROS production and mitochondrial calcium accumulation. So then we asked the question, what are the physiological or pathophysiological conditions in the heart that regulate microRNA 181C transcription followed by translocation into the mitochondria? And so this was the initial thought that microRNA transcribed in the nucleus, like regular microRNA fraction by Drosha, premature microRNA, and then Dicer cleaved it into microRNA 181C that translocate into the mitochondria. And then we also identify certain conditions such as diet-induced obesity, type 2 diabetes, and aging. So diet-induced obesity, 18 weeks of high-fat diet, DBDB mice for type 2 diabetes, and 2-year-old mice. We identify that the expression of microRNA 181C is high, and not only high, we see these overexpression in the mitochondrial fraction of the heart. So this is a very busy slide. A talented postdoc at that time, Barbara Roman. So she identified the precise mechanism during the obesity condition. So diet-induced obesity increased lipid accumulation in the cardiomyocyte, and that cardiomyocyte lipid accumulation activate microRNA 181C expression in the nucleus, and that translocate into the mitochondria, target MTCOX1, generate ROS. That part we already knew, but the connection between ROS and mitochondrial calcium accumulation, Barbara teased out by doing multiple assays where we identify this excessive ROS oxidize a transcription factor called SP1, and that SP1 oxidation causes degradation of SP1. That SP1 degradation reduces the transcription of MEQ1 in the nucleus, and therefore we see less MEQ1 in the mitochondria. And that leads more calcium to get inside into the mitochondrial fraction. So this was the highlight of the mechanism, how microRNA 181C can regulate cardiac function. But how microRNA translocate into the mitochondria? That was the question. I don't think at that time nobody answered. So this is the only hypothesis what we had. Again, we will come back to risk. They are in the cytoplasm. As I mentioned, certain stress can do the thermodynamically distable risk. That's why GW181, 182, they kind of separate from this, and then microRNA and AGO2 complex can translocate into the mitochondrial matrix. We even showed in our initial paper, AGO and microRNA 181C inside the mitochondria, but we did not know how it went. So at that time, there are multiple possibilities in the literature. One of the possibilities after GW cleaved off from this complex, microRNA bound AGO goes through TOM, TEAM, and go directly into the mitochondria. However, there was some skeptical because of the size of these complexes, a little bit bigger than TOM and TEAM pores. So then John Hollander from West Virginia, he identified, or his team identified an enzyme, PNPase. They literally drag microRNA 378 in his case, and AGO2 complex through TOM and TEAM and go inside. However, there is other thoughts at that time that AGO and microRNA go separately. They kind of cleaved off after distable risk and then go independently through TOM and TEAM, go inside the mitochondria. But this requires folding and unfolding mechanism, and how it will work, we were not 100% sure. But what we thought there is another possibility is AGO2 can get stability by its post-translational modification. There are some options, and I'm sure Diego is going to touch on those, so I'm not going to talk about that. And so when we had this picture in my mind, I met this guy in Argentina, and I kind of drag him into this research, and this study is led by Diego. I will let Diego to talk the rest of the study.
Okay, thank you, Sam. As Sam just told us, the obesity induced the overexpression of 181C, which translocate to the mitochondria and target directly empty cuts. This increased the ROS production and mitochondrial production, and downregulate empty cuts and SP1 and MEQ1, ultimately leading to heart failure. But how does the translocation mechanism work? how does the translocation mechanisms work, and even more important, how we can prevent it. To understand the nuclear and mitochondrial communication, I need to go back to GWB182, which is a key protein involved in the formation of RISC, where it interacts with microRNA 181C and EGO2 to upregulate the gene expression, to regulate the gene expression. It's played a crucial role in the microRNA mediated gene silencing by stabilizing the microRNA EGO2 complex, facilitating the repression of the target messenger RNA. However, due to thermodynamic instability between the microRNA interaction, GWB dislocate from the RISC complex, leaving the microRNA and EGO2 free in the cytoplasm. In this condition, microRNA 181C and EGO2 complex can translocate to the mitochondria. Several studies suggest that the phosphorylation of EGO2 in serine 387 in humans or serine 388 in mouse by AKT enhance its interaction with GWB182, and this stabilizes the RISC complex. Under this condition, the translocation of microRNA and EGO2 complex to the mitochondria will be inhibited. So this aligns with our initial hypothesis, which posits that the phosphorylation of EGO2 at serine 387 inhibits the translocation of microRNA 181C into the mitochondria by increasing the cytoplasmatic stability of the RISC complex. Basically, what we say is if it's phosphorylated, it will be more stable and it will be not able to enter to the mitochondria. In the meantime, if it's desphosphorylated, it can translocate to the mitochondria. Initially, we study in vitro the action of AKT using insulin to activate. Insulin is an upstream element of AKT, so we think that activating AKT, insulin, we can activate AKT and it will phosphorylate EGO2. It will make a more stable RISC complex with GWB182, and it will keep outside of the mitochondria. To do this, we isolate neonatal mouse ventricular myocyte, and 24 hours later, we began a short incubation with insulin. We examined the early stage of the insulin cellular signaling pathway by analyzing the phosphorylation of AKT at serine 473 residues and EGO2 at serine 388 residues. The phosphorylation of AKT was significantly increased with insulin treatment. This result was also associated with an increase in the phosphorylation of EGO2 in the NMBMs treated with insulin. That means that this pathway is working. To test our hypothesis that an increase in phospho-EGO2 will enhance on the association of EGO2 with GWB182 in the cytoplasm, we examined the EGO2-GWB182 interaction through immunoprecipitation in insulin-treated NMBM. We found that insulin treatment increased the amount of GWB182 immunoprecipitated with EGO2 antibody. Next, we analyzed the expression of microRNA 181C using qPCR in both NMBM lysate and isolated mitochondria from NMBM. Insulin did not affect the expression of microRNA 181C in NMBM lysate. However, it significantly lowered the microRNA 181C expression in the mitochondrial fraction, suggesting a role of EGO2 phosphorylation in the translocation of microRNA 181C into the mitochondria. One of the final consequences, like some say, of the microRNA 181C translocation in the mitochondria is the dysregulation and accumulation of calcium. Therefore, we measured mitochondrial calcium in live NMBMs using confocal microscopy during treatment with either vehicle control or insulin. To measure the mitochondrial calcium, we transfected the cells with adeno-associated virus with this probe that's called chameleon. This probe is radiometric. The chameleon probe works through a FRET mechanism. When calcium is present, the probe parts come closer together, changing the fluorescence from cyan to green. As you see here, insulin treatment in NMBM result in lower levels of mitochondrial calcium. We have observed that insulin signaling operates acutely rather than chronically, possible due to its negative feedback mechanisms. Basically, we have here this activation, and we can sustain this signal only for a few hours, but it's not able to chronic treatment. That will be possible for a negative feedback mechanism that insulin present. So at this point, we ask ourselves, what's happened with another elements that may activate AKT? Naturally, we consider estradiol as a potential activator of this mechanism in AO2. Once again, to test our hypothesis, we choose to treat human cardiomyocyte cell line AC16 with estradiol for 48 hours to mimic the microRNA 181C upregulation condition, which occurs in the heart in response to obesity. We transfected microRNA 181C into the AC16 cells to the same time to estradiol. We examined the phosphorylation of AKT and AGO2 to Western blot. We observed a significant increase in the phosphorylation levels of AKT and AGO2 following estradiol treatment for 48 hours. As you see here in this panel, you can see the section of the estrogen signaling that is working through AKT and phosphorylate AGO2. That will make this complex more stable. To evaluate whether the phospho-AGO2 decrease microRNA 181C entry into the mitochondria, we isolate the mitochondrial fraction from AC16 cells, and we evaluate the mitochondrial microRNA 181C expression in total cell and mitochondrial fraction. While the estradiol treatment did not change the expression of microRNA 181C in the cells, it significantly decreased the mitochondrial microRNA 181C levels in overexpression. That means the estradiol treatment decreased the expression of microRNA 181C in the mitochondria, or translocation. Next, we analyze the expression of the direct mitochondrial target of microRNA 181C, MTCOTs. Estradiol treatment resulted in significantly upregulation of MTCOTs in both with and without microRNA 181C overexpression. That means this translocation is not happening. MicroRNA 181C overexpression in the mitochondria leads to the degradation of SP1 and ultimate downregulation of MECU1. It's MTCOTs, it's downregulated, so increase the ROS production and increase the oxidation and degradation of SP1. That means downregulation of MECU1, and that will cause the increase on mitochondrial calcium and accumulation of the mitochondrial calcium. So we analyzed by qPCR and Western blot the expression of SP1 and MECU1. The microRNA 181C overexpression result in significantly downregulation of SP1 protein without altering SP1 messenger expression. However, SP1 protein levels were significantly higher in cells treated with estradiol and microRNA overexpression. Here. the microRNA overexpression result in significantly downregulation of protein without altering SP1. Okay. Sorry. On the other hand, microRNA 181C overexpression also decreased the MIQ1 messenger protein expression. But estradiol treatment prevent this decrease. With all this information, the activation of the mechanism in EGO2 by estradiol opened a new dimension for this study. Epidemiological data have shown that premenopausal females had a reduced risk developing cardiovascular disease compared to their male counterparts, where the risk in postmenopausal women match or even exceed the rates of men. So that's bringing to our next question. Are females protected from the consequence of the microRNA 181C translocation into the mitochondria? To answer that question, our initial hypothesis was used to test our initial hypothesis. We use male, age-matched male, female, and overactomized mouse hearts. In overactomized mouse hearts, the lack of estrogen will lead to reduce the activation of AKT and consequently lower the activation of EGO2. Once again, we analyzed the phosphorylation of AKT at serine 473 recitals and EGO2 at serine 388 recitals. Western blot result revealed elevated levels of phospho-AKT in females' hearts compared to male mice. However, ovitectomized hearts exhibit similar expression to male hearts. This result was associated with a significant increase in phospho-EGO2 females compared to males and ovitectomized hearts. To demonstrate that phospho-EGO2 can increase the binding affinity of EGO2 GW182, we use a whole heart lysate and immunoprecipitated with EGO2. We found an increase in the amount of GW182 immunoprecipitated with EGO2 antibody in females' heart compared to male hearts. However, ovitectomized hearts showed decrease in EGO2 GW182 associated compared to females' heart, which is consistent with our hypothesis. We found the levels of microRNA 181C in the mitochondrial from these hearts. Females express a markedly lower levels of microRNA 181C into the mitochondria compared to both male and female heart, ovitectomized female heart. To evaluate the downstream effects of mitochondrial translocation of microRNA 181C in the heart, we measure SP1 and MIQ1 levels by qPCR and Western blot. There was no difference in the messenger levels of SP1. However, SP1 was significantly higher in females' heart compared to both male and ovitectomized heart. Female hearts also show markedly MIQ1 expression, both in messenger and protein levels compared to males and ovitectomized heart. Taken together, these findings indicate that estrogen increases the phosphorylation of EGO2, which leads to a more stable complex or risk complex in the cytoplasm. Consequently, this reduces the microRNA entry to the mitochondria, and which prevents the SP1 degradation and allows the MIQ1 expression. So our next question was, does estrogen protect heart from obesity-induced damage by EGO2 phosphorylation? So our previous study demonstrated that male mice fed for 18 weeks high-fat diet developed cardiac hypertrophy by upregulated microRNA 181C in the mitochondria. To determine whether there are differences in the development of cardiac hypertrophy between sexes, we randomly switched 7 weeks old male and female mice to a high-fat diet for 18 weeks. We measure hair size measured by the ratio of heart weight and tibia length, and we observed that female heart size was significantly smaller compared to male heart, when both were fed with a high-fat diet. Additionally, we analyzed two molecular hypertrophic cardiac markers, B-type natriuretic peptide, BNP, here, and beta-myosin heavy change here. No difference were detected in normal chow condition between sets. However, in high-fat diet, the high-fat diet induced the expression of these markers in males, while in females, we did not see the response of this induction. We propose that higher phosphorylation of AKT levels in females' heart will limit the entry of microRNA 181C into the mitochondria, preserving empty COTS levels. Female hearts showed a significant increase in phosphorylation of AKT compared to male heart, both in 18 weeks of normal chow and high-fat diet. High-fat diet significantly reduced the phosphorylation levels in male heart, but not in female heart. In male mice, high-fat diet led to significantly downregulate empty COTS level in the heart, while not such downregulation was observed in female mice fed with a high-fat diet. Finally, we analyzed the mitochondrial expression of microRNA 181C. Consistent with our previous finding, there was a significant increase in microRNA 181C expression in mitochondrial fraction after 18 weeks of high-fat diet compared to more normal chows in male mice, where mitochondrial microRNA 181C expression remains unchanged in female heart fed with a high-fat diet. So the conclusions from this study are: estradiol was found to be a key mediator of EGO2 phosphorylation by AKT activation, which inhibit the microRNA 181C entry into the mitochondria. We have identified an important molecular mechanism where the estrogen depends phosphorylation of EGO2, which increase the stability of risk in the cytoplasm of cardiomyocyte and significantly decrease the microRNA 181C levels in the mitochondrial fraction, limiting the subsequent downstream detrimental effect. And this mechanism could be important, providing female-specific protection against obesity, cardiac hypertrophy, in contrast to age-matched males. So our next question was, can the activation of EGO2 per se prevent the translocation of microRNA 181C to the mitochondria? To answer that question, we have now generated a transgenic mouse model developed in our laboratory using CRISPR-Cas technology, in which the serine 388 in EGO2 is mutated by aspartate. This model allows to study an hyperactivation of EGO2. Basically, we change this serine for aspartate, a huge group with a negative charge that will mimic the hyperactivation of EGO2 or the phosphorylation of EGO2. As our first step in this project, we extracted the heart from wild type and EGO2 mice. We then isolate the mitochondrial fraction of this heart and extracted the messenger RNA to create, or RNA, total RNA, to create a microRNA library. And we measure the copies per nanogram of microRNA 181C by digital PCR. We found several microRNA that were either upregulated or downregulated in the mitochondria of these animals, one of which was microRNA 181C, which showed almost a threefold decrease in AGO2 mice compared to wild-type mice. Also, we measured the copies per nanogram of microRNA 181C using digital PCR in the whole heart, cytoplasmic fraction, and mitochondrial fraction from the wild-type and AGO2 mice. Here we show in whole heart and cytoplasmic fraction. And as you see, we did not see significant difference between wild-type and AGO2 mice in whole heart or in the cytoplasmic fraction. However, the mitochondrial from hearts of AGO2 mice have significantly lower microRNA 181C expression compared to those from wild-type mice hearts. So I hope to share more results with this model. And I would like to thanks to all the authors and all the audience. And also I would like to thanks to Dr. Vagnozzi and Dr. Tish Murphy. Thank you.
Thank you very much. That was a very, very interesting and pretty comprehensive study. A lot of work and congratulations. Let's see, if people have questions, you can put them in the Q&A or the chat. And I'll start with the questions that are here. Caroline Gonzalez asked, are there differences in and she asked this in the middle of the talk, so you may have answered some of this later, further on in your talk, but are there differences in microRNA 181C expression between male and female in healthy heart versus injured heart?
Sorry, can you repeat?
Sure. Are there differences in microRNA 181C expression between female and male hearts in healthy versus injured hearts? I think you did show some
Yes.
data on this.
Yeah,
I think
we
this
actually
question was asked
showed
before. Yeah.
No.
So you just
Here. Yeah.
just
No.
share this. They cannot see the screen.
Oh, okay. Sorry. Let me share the screen.
That's the answer.
So, okay.
Mitochondria and then.
Yeah. So, difference between heart, like here in female heart, we did not see difference between normal chow and high fat diet.
Yeah, so under normal condition, we see a statistical significant difference in the female heart. Oops.
Yeah, go back to where you were. Yeah.
Yeah.
One question that I had is, so this is in the mitochondria. Are there differences in total?
Yeah. Go ahead.
No.
Yeah, total is also the same. As our initial paper suggests, the microRNA 181C mainly localized in the mitochondrial fraction. So very few expression we detect in the cytoplasm. It was so yeah. So I mean, total heart and mitochondrial extract will kind of the same in terms of the expression of 181C.
Okay.
So yeah, the normal condition, as you see, in the female, we have less microRNA 181C expression. With high-fat diet, as Barbara's paper suggests, that lipid accumulation activate the transcription of 181C. We see both upregulation, but in case of male, the blue bar graph suggests a statistical significant increase from the normal chow. However, in female heart, we do not see, we see increase, but not statistical significant.
Okay. All right. Thank you. There's another question from Dr. Tomar. Great work, Sam and Diego. Thanks for sharing. AKT is known to translocate to the mitochondria. Do you think this translocation could be influenced by estradiol? Additionally, how might it affect the translocation of microRNA 181C? They have two questions, so I'll let you do the first one first. So AKT can also translocate. Do you think that could influence your data?
You want to answer? I mean, he knows way more than I do, but he likes me to talk. So, Dr. Thomar, thank you for the question. Yes, it is indeed possible that AKT, when it gets phosphorylated, go inside the mitochondria. However, we did not look at the phospho-AKT in the mitochondrial fraction. The reason is we are more focused on the risk stability in the cytoplasm. So we are focused on how AKT can influence the post-transcriptional modification of AGO2 that will increase the stability. So if AKT goes inside the mitochondria, we cannot study that. I mean, there may be a part of AKT gets phosphorylated and go to mitochondria, but we don't believe that will have any influence in the cytoplasmic risk stability. So unfortunately, we should have looked at it, but we did not. But hopefully that will answer your question.
Always more to do. And
Yeah.
and there's a second question they have, which is you showed drastic changes in MICU1 levels between males and females. Are there differences in mitochondrial calcium content or flux between the sexes?
Good question. Obviously, we did not. That is a plan that we will check the mitochondria. What we did is phospho-PDH level. We did, like, as we know, it's a surrogate marker of mitochondrial calcium. And so we looked at the phospho-PDH level when we are saying that MICU goes down and calcium goes up. We looked at the phospho-PDH expression, and that is how we measure, but we did not measure directly calcium, and that is what we are planning to do in our new mouse model.
Let me ask a question. I mean, MICU1 is complicated because it's kind of has two modes of action. If you, and again, knockouts maybe, but if you decrease the amount, you increase calcium uptake at low calcium levels. That's kind of its threshold. But then at high levels, it has the opposite effect. And so the question is, do you have any, I mean, and that might alter things like, calcium activation of dehydrogenases may be less activated if MICU1 is higher. So have you thought at all about how it may depend upon what the cytosolic calcium levels are, what the effects of MICU1 are?
Yes, that's exactly what we are currently working on. Yes, so as you pointed out, if cytoplasmic calcium level is low, MICU1 low means a higher mitochondrial calcium, and if we activate or some stress condition such as heart failure and all, like TAC model, if the cytoplasmic calcium level is high, that's what we are trying to do. We'll TAC some of this mouse model, and hopefully in next webinar, we can answer your question. If the cytoplasmic calcium is high, what will happen?
Sounds great. So there are lots of questions. There's an anonymous attendee who asked, they say, very interesting and great work, which I think everyone agrees. This is Elena from Florence. I did not catch how the cell discriminates between microRNAs that need to go into the mitochondria, how the complex chooses which microRNA carrier versus mitochondria, chooses microRNAs to get into the mitochondria. So how does the, I mean, it's kind of a big picture question. How does the cell decide which microRNAs go in?
It's a thermodynamical decision, basically.
Yeah, so there is a review on this. Obviously, we did not do any of those bioinformatics prediction and all. So there's a review paper in Cell Metabolism in 2017. They said that AGO2 and GW182, when they form a risk complex, there is a thermodynamic stability minus 30 kilodalton or less. If that is the condition, then it will be a stable complex. And then if somehow we lost the stability, like the way Diego showed, by phosphorylation, by lipid accumulation and all, they find the target. So if there is no target, of microRNA 181C based on the stability factors. Again, don't ask me follow-up question because my knowledge goes up to this. We cannot go stretch it. But yes, if the stability somehow between 3' UTR and microRNA goes below the threshold, then they cleaved off with AGO2, and then they kind of make a PUE complex and go different fraction of the cellular fraction, such as mitochondria, SR. And if there is no target, even in these components, either they go degradation or they go secretion outside the cell. So the thermodynamic stability is the answer between the microRNA messenger RNA binding and selection, whether it will go to mitochondria or it will go outside the cell.
Very interesting and very complicated. And as you pointed out in the beginning, I mean, for DNA that was considered junk, it's come a long way. There's another question, and I apologize if I pronounce your name wrong, from Dr. Chattopadhyay. Sorry, I'm sure I did not pronounce that correctly. But I have a question. In human physiological stress conditions, mitochondrial cyclooxygenase 1 copy number increases, which increases the ROS as well. Can you please explain why here it is decreasing, just like physiological disorders, MDD and Alzheimer's? So I think the question, as I understand it, and the author can jump in if I'm not asking it right, but basically I think they're asking, in some stress models, copy number increases. Here you're finding it decreases. And what are your thoughts on?
Again, this will be a hypothesis. We do not have any data of those pathological condition like Alzheimer and all. But what we are saying, that when microRNA 181C in the mitochondria goes up, it downregulates the MTCOX1 protein, not the copy number. So this is a translational inhibition. And because of the less MTCOX1, the complex 4 has some kind of remodeling. Well, and that protein complex in complex 4 makes, Dr. Steenbergen always say there's a water flow, and if you block it, the water goes, and then you put pressure in complex 3, and ROS is produced. So we do not see any messenger RNA of MTCOX1 upregulation or downregulation. So I think we are focusing only on the protein, not copy number of the gene.
But I think, the question is, even if you just look at the protein level, I think, there are many ways to, or not many, but multiple ways to make ROS. And if the electron transport chain becomes, unbalanced, there may be multiple ways. But I think it's an interesting question and something to think about. Dr. Holker says, wonderful session, congratulations and thanks. And then there's a question from Moni Nadar, Nader. Is phosphorylation of AKD different between genders?
You have that data.
Yes, actually it's true. In male mice, it's significantly decreased compared to female mice in both conditions, high-fat diet and normal chow.
And that's consistent with your model. Are there any other questions? I think that's all that's in the chat, right? That's all that's in the question and answer, and I don't see anything in the chat. If not, we're coming in on the end of the hour, and this, I think, was a wonderful session, a lot of work, and I'm very happy to see people looking at sex differences in cardiovascular disease. I think that's an important topic, and I congratulate both of you in your lab for adding to this very interesting literature. If there are no further questions, there's one other. There's just more congratulations. And so I think if there's nothing else, do you have any closing comments, Sam or Diego?
I would like to thank you for your time and moderating the session such a nice way, and Ron for organizing it with Jenny, and obviously JMCC for giving us this opportunity.
Yeah.
Ron, do you have any closing remarks?
I'll just echo the thanks and congrats on a great presentation. Also, this recording will be on our YouTube page. There's a recurring JMCC webinar recording page if you want to revisit this, or if you have colleagues who haven't seen it. So please check that out. And I think that will close our session. Thanks, everyone.
Diego,
Thank
did you have any
you.
closing remarks? Did I? Anyhow, it was a great session, and thanks everyone. This was very informative.
Thank you so much.
Thank you.
All right.
Good Lord.